In this video, we'll explore a case study of a potential application of Tropolink, unraveling dissemination trajectories of an insect vector of plant pathogens. In the context of plant health, vector-borne pathogens cause tremendous crop losses each year, underscoring the importance of better understanding epidemic processes. In this case study, we'll use Tropolink to try to identify the likely dissemination trajectories and patterns of an insect vector of plant pathogens. Here, Cacopsylla pruni, a jumping plant lice transmitting Candidatus phytoplasma prunorum, a phytoplasm causing the European stone fruit yellows, a disease affecting wild and cultivated prunus trees like apricot and plum trees. Let's take Montpellier's site for example. Leveraging our knowledge of the population dynamic and life cycle of the insect vector in this region, we'll compute backward air mass trajectories corresponding to possible arrival flights of the psyllid in orchards in spring using Tropolink. Here, input parameters for Tropolink include vector arrival dates and site, extracted from historical vector abundance data, flight duration and arrival hours, extracted from the literature and observation of the insects, and arrival altitude, determined through study of the height of the planetary boundary layer at the arrival site, a parameter available in Tropolink trajectory outputs. Then, once the study has finished running, we extract the trajectory output from Tropolink and import them in R, the software we will perform all the following data analysis. This gives us all possible air masses used by psyllids when arriving in Montpellier each spring. Then, we filter these trajectories using meteorological data available in the trajectory outputs given by Tropolink to keep only the trajectories wherein psyllid could realistically fly and survive. Thus, we obtain the air mass trajectories corresponding to all possible arrival flights of the psyllids the closest to their biology and atmospheric physics as possible. From these filtered trajectories, we can then compute the connectivities between the arrival site of the insect and each cell of a grid covering France using the code for connectivity computation in R available in Tropolink Wiki. Thus, creating a heat map of potential origin of the psyllid before the arrival flight in spring, where they can infect fruit trees. Then, comparing this heat map with distribution of the psyllid overwintering host plant, we can determine precise potential source site of the psyllid, which we can then sample in a targeted way to confirm or not our hypothesis via population genetics, and, in fine, identify the true sources of the vectors before the arrival in our orchards. The main prospect for this work is to inform the development of new surveillance and prophylactic strategies aligning with the global imperative to reduce pesticides use in plant health.